Hello and happy Monday. I still like saying happy Monday. I kind of take weekends, so it still feels like a Monday to me, which is good because I actually do pretty well on Mondays because I've had a little bit of brain rest where I haven't done work. Usually by like Tuesday or Wednesday, my brain is like, oh my God, you have to do the same thing in a row again. So I like Mondays. It makes my being able to do something new and different from what I did yesterday makes my ADHD brain a little bit happier. I need to find a way to do that on like a Wednesday so I can recreate Monday on Thursday. That's a good idea. I'm going to work on that for the future. Okay, this week, my study skill note is going to be about studying for exams because we are getting so close to the end of the semester that that's probably the thing most people are going to be doing. And so that's what I'm going to talk about all week is exam preparation, and especially exam preparation in this kind of situation where you're probably taking them somehow at home in some manner. So I don't know exactly what your instructors are going to do. I know that my class, all my exams are online already, so they already do their exams this way. And therefore, if your exams are online in some manner, things are in some manner open book. That's actually kind of important because of the way it's done. Now, there are some people who might still be doing like you have to be on video while you take your exam, so you can't do that, but I'm really hoping most of our instructors choose a whole different route. So I'm going to pretend you are now taking online open book exams and treat it like that. So if you're taking online open book exams, what are the key things you need to do? Well, first of all, you need to prep in various ways. Just because an exam is open book does not mean you can't prep. So for essentially at least 3 of the 4 days I'll talk about this week, I'm probably still going to talk about studying for and prepping for exams in different ways. So today, I'm going to start by kind of bouncing off of where we were on reading last week by talking about recall methods, because recall methods are really important. They are still the thing that gets stuff stuck in your brain. So even if you're taking an online exam, there's a strong chance it's going to have a timer of some kind on it. I put a timer on mine. I think that's important because I do want the students to have the information in their heads. If you have to look up every word in the exam, it's going to take too long. Some of the information has to be here and ready to go, which is also just important for, being able to follow material you've learned. So if you were using reading techniques in which you were practicing recall, you are probably doing some of these things already, but let's go over how you would use some recall methods to study for exams. So the very first thing is that you would not, under any circumstances, just reread the material you've read before. Don't do that. It's a bad method. It's actually one of the worst methods. It's up there with highlighting. Also, if you highlighted your material, definitely don't reread the highlighted segments. This also doesn't work because, one, it doesn't work. It doesn't actually put the stuff in your brain. It's still just kind of going to your eyes and maybe your working memory, that part that goes away quickly, but it's not cementing. But because it goes into the working memory and doesn't fully cement, it makes you feel like you kind of know it. And then when you see it again tomorrow, if you reread the same thing or like 2 days later, it makes you feel like you kind of know it. And you start to feel pretty good about it. Like, hey, I kind of know this. I remember reading it the next time when I read it the third time. But the problem is that you don't know it. You just recognize it. Your brain loves to recognize things. It's like, I know that thing. I feel good about that. But if you look away from it and you can't say that information or you can't provide it without it being in front of you, you're not doing what you need for a test, but you're making yourself feel good about it. And I don't like that because it makes people overconfident. It's like, no, but I read my chapter like 3 times. How did I do poorly on this test? Well, because you weren't practicing the skill you needed for the test, which was not to vaguely recognize things, but to actually recall or pull from your long-term memory how they sort together. Sometimes, if you're taking poorly written multiple choice tests, recognition will get you by. If you know that's what your instructor does, hey, you might be good, because then you'll recognize the first part of the multiple choice, you'll kind of recognize the second part of the multiple choice, and maybe you'll be able to put those 2 pieces together. I write very good multiple choice tests in that the answer choices are similar enough or related enough, or you have to use enough critical thinking that you can't just recognize them. They all will look the same, or 2 or 3 of them will look like they belong. So I already know you can't use that method for my tests, and I hope other people are doing similar things because you got to practice the process. So what can you do? Well, here's where you need to do the same thing with the reading, which is first make a plan. You know what you need to know. If you need to know terms, then you need to be practicing words and definitions, either using flashcards or a blank word sheet or something like that, where you look at a word and you know its definition, or you look at a definition and you know the word and you can go back and forth without looking at anything. My chihuahua is trying to escape from a cave here. You okay? You stay there. Okay. He's all right, I think. So flashcards and that kind of stuff are important. If what you're going to need are steps to something, then you need to make sure that whatever the steps are, that you are able to repeat them in order, or you have a device to help you remember them. Mnemonics are one of my favorite things. So that'll be the next thing that I talk about. Maybe I'll spend time tomorrow on mnemonics. Note to self, tomorrow, mnemonics. Mnemonics. Yeah, it's a really hard word to spell. Nix. Okay. I don't spell particularly well. Students ask me all the time, can you spell that? And the answer is no. So you need to know what you're looking for. Do you need to know the steps? Do you need to know concepts? Do you need to know how to classify or categorize things? That's really common in my biology classes. Do you need to know things that happened? If you're talking about history, you might need to know important names, important dates. They work like terms. You got to be able to back relate them to each other. So then that's the information you need. So the pieces you need to know, you have to practice using a recall method. So using flashcards, using quizzing yourself. Make up a quiz of the things you think you're going to have and quiz yourself a bunch of times. It's a really great method to do that. These are the easy things you can do. Or even do the reading techniques where you go back to your chapter and you kind of scan through, or maybe you see a heading and your goal is to remember some of what's in there. And then once you try to remember it, then you can go back and check yourself. So any of those methods for getting down sort of the basic information that you need. If you have the option of getting together with other people, online, whatever, quiz each other. Have each other make up a bunch of questions and then share them. You can do that without even having to be in the same place because you can share questions with people. You can get together in a digital study group and share questions. Hopefully really good instructors are having students do that. I should start adding actually that to some of my online classes. That's a really good idea. I'm going to make that note. Students share questions in study groups. I have a lot of cool things to add to my online class for next year now. Actually, for this summer now. So that's one of the key things. And then try to think of more difficult questions. Try to do some free write things with larger questions. So if in biology, I know all my examples are biology, a question is about a process, then you might say, well, if I have to know this process, if I was given a question about, could I just free write the entire process? That's a recall method. It's good to practice that. That's something you can do. You can, I'm trying to think of all my favorite recall type methods. So same thing might work for something like psychology. You have to categorize or put together all the steps of something. So that would work in that kind of case. In the case of problem solving, solve problems. Find new problems and solve them. In the case of math, Khan Academy has tons of that kind of stuff. You can definitely go find them. Usually finding practice problems is not that hard. If they're the kind of thing that you can kind of fake make up, then do it. Even if they don't work perfectly, at least if you find the steps to it, you're practicing. So those are all of my starting point for studying for exam methods. So tomorrow I'll talk about the fun versions of recall, stuff you can do that really gets your brain going and is a little bit more exciting. Stuff I've had to do, because let me tell you, memorizing shit is boring. And ADHD brains don't do boring. So I've had to learn a lot of different methods. Okay. For today's COVID science, I am actually going to start at the very beginning of something, as in a friend sent me a news article and I have actually barely looked at it. I scanned through the title. So I'm going to show you what I do when I look through a news article. So that's my starting point. So the news article is from The Hill, which is not a bad reporting source, but is still a commercial-based reporting source, and they are certainly not. I have seen many a clickbait headline on them, so it's not like they don't have it. And in this case, the headline is "Coronavirus Detected on Air Pollution Particles Report." Okay, so I'll tell you what I'm thinking. Problem one. First of all, as a reminder, anytime we're looking at any article, detecting virus and detecting active virus particles that can make somebody sick are two completely different concepts and should always be thought of that way. DNA and RNA last a really long time in various places. In fact, and we've gotten really good at detecting really tiny amounts of them, which is great. I love that science can do that. For example, when people are in very big disasters and we can't find bodies and things, we can detect, or they're like ruined beyond repair, we can detect their DNA from such small pieces that we can identify them. That's lovely. But again, those are not live people or other things. We can also detect from like tiny little swabs so we can match DNA. This is great. But in the cases of viruses, it doesn't provide us with much information other than there was a viral particle there, maybe. And that's really a maybe, because really there was only viral RNA there. The particles could have already been destroyed by the time the RNA got there. So whenever I see a headline like that, that's always the first thing I think. So as I scan through the post, or the article, it notes that it's in Italy. So this is a place that at least has had a lot of this kind of stuff detected on particles of air pollution. It was reported actually not originally by The Hill, but by The Guardian. I always love it when news sources cite other news sources, but And The Guardian is also a good paper, but also not beyond the capability of not getting their science right. So the easy version of this, it says they collected outdoor air pollution samples at urban and industrial sites, identified a gene specific to COVID in several samples. Oh, I definitely am going to need to know a lot more about that. It does have a link to the study, so we will get to that in a moment. It does note, and this is the important line in the article, thank you, The Hill, for including it, and including it as the third paragraph. So I will give them a little credit for this. The study has not yet been peer-reviewed. It does not determine whether the virus and pollution particles is in quantities sufficient enough to cause the disease in humans. Of course, I think what that means is that this information is utterly useless to regular humans and therefore is not news in any way, shape, or form, and should not be reported in any way, shape, or form. But I am apparently not in charge of the news, so I don't get to say that. Then it talks about some of the things that came into this, confirmed in an independent laboratory. They talk to a researcher, and it notes how there's a lack of information about how the disease can be carried by pollutants, which is obviously because we only just started studying things. And he says, actually, I really like this line. I'm a scientist and I'm worried when I don't know, which I would hope every scientist was. That's like the whole thing of being a scientist is, I don't know how that works. Can we find out how that works? Let's figure out how that works. And that's important. We should always, as scientists, be trying to figure out how things work. But we should also be very aware that we don't necessarily have all the answers in one study to those things, or are going to be able to solve them, or that other regular people should be worried about these things. We're scientists. I just want to know how things work. Most scientists just want to know how things work. It doesn't mean that we're looking at this because we think it's the major way this virus is transmitted. In fact, I can tell you already from what I've read that that's not true. So when we quote scientists like that also makes me worry, because the media is like, this guy's worried. And it's like, well, he's a scientist. He should be. It's like when the scientists are saying right now that we don't know if antibodies confer immunity to this. That's accurate. We don't know. Can we make some implications based on lots of previous data and things like that? Sure, but will we ever say we know? No good scientist worth their salt will, so don't let them. All right. And then they start into the suggestive stuff. If it was able to grab onto a place, it could say that polluted areas could be more susceptible. Other research groups have suggested this could help it travel. This is where everything starts to kind of be like, oh man. And then it notes that the northern part of the country, which is most affected, is also most polluted, which again is a correlation, meaning the two things happened at the same time, but it doesn't mean anything useful. So here's that information. Let's go look at the actual study, because I far prefer actual studies. And again, since this study is not yet peer reviewed, I am essentially going to peer review it for you, or at least I'm going to do it like I would as a peer reviewer, which I think is important. All right, so I'm going to examine this paper. So there's a lot of people involved in it from a lot of different places, but mostly in Italy, so that's good. Let's see. It notes the basics that we already know: spread by droplets, spread by contact, that this part of Italy was hit very hard, and that these are the results that they performed on 34 PM10 samples of outdoor airborne PM10 from an industrial site, compared with collected with 2 air samplers. Okay, we haven't even gotten very far, and I gotta say, 34 airborne samples may not be a lot. And also, collected with air samplers makes me wonder a little bit about how these were sorted and put together. So let's see. They do explain it with methodology that they use a gravimetric air sampler. So they're taking a chunk of air, so 38.3 liters per minute for 23 hours. So a lot of air goes through these things, like way more than you are going to go through walking, just to be clear. And they trap it onto filters with some retentions. They're trying to trap particles. It doesn't say the size of these filters, but I'm sure it's got it somewhere. Oh. I'll figure it out. So they push a lot of air through a sampler and catch it on a filter. So just be aware of that. And then they take those filters and they remove the stuff from them, and they look for genes, which again, we're real good at. Our PCR skills are amazing. We can find genes in teeny tiny little places. So doesn't mean a lot. Let's see. And then, oh, this is a great line. They talk about a bunch of other stuff, but one of the things says to avoid running out of the scarce sampling material available, which tells me that the reality here is that it's pretty hard to air sample, and it takes a lot of air to sample. So this is already interesting, but not interesting from a I'm walking outside perspective. Because otherwise, if you could just take air samples, you would just stand out there and take air samples. It wouldn't take that long. So, and due to a lack of filter materials from the filters, they were not able to repeat enough the test to show positively for all of the molecular markers simultaneously. There wasn't enough genetic material, period, to run enough samples to do enough of this. Not gonna lie, I already have problems with this. As a scientific paper, I would already write back and be like, I'm sorry, what? Why didn't we do more? So at best, letter to the editor with preliminary results, and therefore everything associated with it should say preliminary, or even better, sort of pilot study, meaning that there's not really enough data here, but we gave it a try. And that's not what it said. It said coronavirus detected on air pollution particles. This is definitely problematic. And really, it says this is the first preliminary evidence that it can be present on outdoor particulate matter. They're not even talking about amounts. They're just talking about the fact that they detected it, and it doesn't even sound like they detected a lot of it. No. Okay, so they actually give, let's see, all 34 of them, and they detected some genes in some cases, and in some cases only one of the genes, or only 2 of them, and it is hard to tell which ones they did. And it goes back and forth. So with a lot of data collection, there might be a little bit of this in the air collected in an area that has had a lot of this. So from a scientific perspective, this paper has preliminary data problems. You really can't just have a thing where you say, well, we didn't do all the analyses on all the pieces of data. That is not actually going to fly for a full scientific article. So that's item 1. If they want to do all 3 tests, they need to do all 3. 34 samples is very low, and being able to not even be able to get more samples is problematic. And they were in some very specific locations, and they don't really describe it very well. So this is, and it is indeed, marked as a letter to the editor, which makes it not a full scientific study. In science, we have multiple things. We have full studies that provide sufficient data to make conclusions, and we have letters and preliminary studies, which only provide sufficient information to form a new hypothesis. It is never considered to be complete data. So that's really the key to this, is that this doesn't answer a lot of questions. It creates a bunch of questions. What are these things? What are we finding? Is there a lot of it? Could we find better ways to collect it? Is it meaningful? Are these anywhere near particles, or are they not particles? And all of these have no answers. So, so far, it would appear that we're finding RNA on these things. And that's about it. There's not a lot else here. So do I think that either the Guardian or the Hill should have reported on this study? Not even a little bit. This is far from the point of science that we should be talking about, at least in the media, because this is the point of science that scientists should be talking about. Is it useful to a regular human being? Probably not at all. Does it even tell us that being in a polluted area like this could be good or bad? Not at all. So that's how I feel about it. And if I was reviewing it, I would definitely send it back with a number of different comments, asking them to be a little bit more chill. But I mean, the scientific paper is even more chill than the other thing, the Hill article. But I also dislike it when the scientists involved even give commentary on this kind of stuff. But also, I just wish we had a whole media system in which we didn't do that without having an understanding of what's good science, what's bad science, what's preliminary, how it all works, and really explaining that stuff to people as the first 3 or 4 paragraphs, instead of saying, oh, we found virus on pollution particles, because now everybody's going to be worried about everywhere they go. Don't be. It's still not a thing. And based on the amount of air they had to get the small amounts of RNA they had to get in even just some, not even all, some of the samples, tells me that there's not a lot of this in the air, even in a place that was pretty well affected and probably would have had good air retention. So that makes me feel even better about going outside. Cool. So that is my general scientific opinion of this article. I'll leave the links in the comments. I'll be back tomorrow with more information.